So now let's take a look at some of the slides we made, both from our mouth, direct M, as well as from our broth and our plate from the Aseptic Techniques lab. So I've already got one of them on there, and I'm not going to tell you which is which because I just want to focus on what we're seeing. Okay, so first of all, I'm at the 4x objective, and when we look at the screen, I think that's pretty cool looking. I mean, all that purple, all that darkness, very neat. All these shards around here, very cool looking, but totally not what we're looking for right now. Okay, so this right here is a big chunk of dye to some material. All of this, well, this one is from my mouth, and so all of these are actually fibers from the swab that I used to grab the sample. And so that's what that looks like. So don't think that that looks like don't think that that's bacteria. So I'm going to go ahead and just kind of move around. For example, that right there, very cool looking, total garbage though. Okay, so don't be taking a picture of that and submitting it. When you're looking around, if you're thinking, well, okay, well, what about these types of things? Well, this is direct M, and so we should be seeing some human cells, some Andy cells. That's me. And so that right here, it might not be as easy to see on the screen, but when I'm looking at it through the objective, I can totally see that they're my cells. So let's go ahead and move to the next objective. And we'll just kind of go in closer. And so what we see here, again, is pretty much just a bunch of garbage. Okay, so again, don't be taking pictures of that and saying, oh yeah, well that's bacteria and that's bacteria. It's not. These are fibers. Some of these things may be bacteria, but it's just not good enough. So over here, what we're starting to see is a human cell. So I'm going to go ahead and put in the middle a cell that should be a little bit clearer to understand what's going on. And so there we go. So hopefully what you can see is, for example, that right there is the nucleus, and the cell around it. And that right there is the nucleus inside the cell. So those are examples of cells that are mine. And then we have a whole bunch of shards all over them, the skinny things. But if we look closely at some of these dots, and that's where I've got a little bit of an advantage here, right? So when I look at, for example, those dots right there, right? So that and this, those to me look like they're bacteria stuck on your cells. Now, how would you know that, right? I mean, I'm telling you that this is garbage, that's garbage, this is garbage, this is garbage. How are you supposed to figure out that that's not garbage? Well, that's what the video is for, right? To kind of clue you in on it. Now, this is a particularly bad slide, but sometimes bad slides are very instructive. I've prepared plenty of slides where I didn't have so many fibers, and if the stain didn't stick so well, what we would see essentially is a lot more bacteria. So I'm just kind of hunting around, just like you would do, right? Looking, a lot of garbage, although it looks cool. And I'm looking for a human cell that will show me some bacteria that are a lot more clear. And this one actually in the middle now, this is a good example where I'm pretty sure that all of these are individual cells. Okay, so for example, this right here, this image, if you were to take a picture of this and upload it and go ahead and put an arrow to the nucleus, things like that, that would be great. So that's an example of direct M on how to find your human cells and also not submitting garbage, right? Nobody wants to submit garbage. So now let's look at a couple of the bacterial slides. And so first, actually, what I'll show you is on this particular slide, after staining, if we get in nice and close, look at the difference between them, right? And so this is P for plate and B for broth, right? It's actually like that, but we know that the bacteria are actually on the other side. And so remember, from the plate, we put a big smear from a colony. That's why we see so much bacterial cell. And here, we see, by the camera, we see maybe a faint purple. I can see it with my eye. So that means that there's a whole lot of material on this and not as much there, but I bet there's still going to be a lot of bacteria here. So let's go ahead and check it out. Let's look at the plate one first, because we know that that is chock full of bacteria. And so when we look at it and we look at the screen, it kind of almost looks like a tissue sample where there's individual different types of tissues, but really this is just a whole bunch of stain. And so what I'm doing is I'm going to area where if you look at here, okay, now this right here, this dark area is actually, see this? I'm changing the focus right now. This is actually the Sharpie mark. And so if we kind of go over here, this is the border of the Sharpie. So when I drew the circle on the slide, that's it right there. And so when we focus in on it, we can also see, that there are blotches of dye. Okay, so don't get tricked into that. That whole thing is your Sharpie. And investigate your Sharpie as much as you want, but don't submit pictures of Sharpie to the class. Okay, so this right here, I think we're gonna have something good here. So I'm gonna go from 4x to 10x, get it in focus, from 10x to 40x, get it into focus, and then I'm going to go to 100x. So this is now 1,000 times magnified, and it's really dark. So what I'm going to do is I'm going to open up my iris diaphragm down here, right? So I'm going to open that up, and when I do, we can now look at the screen, right? So we can see that now it's really bright. So I'm going to close it up, just to kind of remind you, if something is dark to begin with, go ahead and move the iris diaphragm. Okay, and so I'll get that in focus. So now that's really not very focused at all. So what do I need to do? I'm using the 100x objective, so I need to use this, right, the immersion oil. So again, as a reminder, what you want to do for the immersion oil is move the oil immersion lens out of the way, right, and then put a drop right on the slide itself, and then slide the lens right into it. And you may want to go back and forth a couple times just to make sure you do have it in there. And then if we look at the screen again, hopefully when I get this in focus, things will be a lot sharper. Yeah, look at that. Haha, almost there. Almost there. Oh, it's so beautiful. Let's go ahead and zoom in on that. So I'm tweaking the fine focus now. All right, so I was unhappy with the rods, right, the bacillus form in the prepared slide. And just a little bit of tweaking goes a long way. I mean, with my eyes, I can see this quite well, so just bear with me. It seems to have a little bit of a lag on the screen. And I know that you also will be dealing with this when you're trying to get your microscope. There we go. Oh, just a little bit better. All right, well, we're going to leave it like that. Let's zoom out just a little bit. Okay, I can't control myself. I need to try to focus it just a little. That's good. Patience is a good virtue or a skill of a microbiologist. So when I look at this through my microscope, it looks great. Here, it looks fine. This would be a great image if you were to submit this. So what we're seeing then is essentially this right here is a cell. And it's attached to this cell. So this is one cell, two cell. That's a cell, that's a cell, that's a cell. All right? So this right here is a mixture of mainly bacillus, so the elongated bacillus. And there are some coccus mixed in. And that's what will happen sometimes when you have not a really, really pure sample. So I'm just going to go ahead and move this to another zone to see if we can see some individual cells a little bit better. And one of the issues we have here is that this sample took up so much stain that it's hard to see the individual cells that are right next to each other. Okay, so if we look at this then, we see we have a bacillus cell that way, right? Here and here. We've got one here. We've got one there. And what it looks like to my eye, it's really quite interesting. If you look at these circles, they're all lined up. These are either coccus cells. That's exactly what coccus looks like. So if you're looking at a sample and you see all that, that would be coccus, because they're all a bunch of, there's a circle, there's a circle, there's a circle. But I'm also wondering if, due to the plane of focus, if these are little rods sticking out like this. So you see how if this is a rod, if we were looking at it upside down, it'd look like a circle. I'm wondering if that's what these are. But regardless, this is a sample of what bacillus bacteria should look like. And so, yay, I like that. And if you send me an image that looks just like that, then I will know that when you were looking at it under the microscope, it looked even better. So that's the benefit of me using the same system that you have. And when I see, okay, I can get that quality of an image, but when I look at it in the microscope, it's right on. You submit one of those images like that, I'll say, okay, I see what's going on, but I know that the actual microscopy was spot on and you did an excellent job. Okay? So what we're going to do is, now that we've seen bacillus from one of the samples, let's go ahead and take a look at the other sample on this, which is from broth. Remember, what we were just looking at was from my plate sample from the Ubiquity lab. And so if we look at the screen here, what we see is that it's a lot less. I put it back to 4x objective. There's hardly anything there because, again, it was from broth, so there was a lot less material. So I'm going to zoom in and go to 100x. All right. And that was the 10x objective, excuse me, with the full 100x magnification. Now the 40x objective. All right, get this kind of centered. And now the 100x objective. All right, so now what I'm going to go ahead and do is I'm going to go ahead and stick the immersion oil on the slide, all right, as we do. Put the 100x objective right into it. And then try to get it into focus. And so this is an example. This particular slide is from my unknown. And what I can see is that what we have is essentially, we've got these kind of short rods going around. So nothing, you don't see a bunch of circles and you don't see a bunch of rods like the one we just saw, but the fact that it's like this, it's like this, it's like this, it's like this, those are indicating that those are rods. And under the microscope, I can see it a lot better. These shards here are, I think those are just some kind of artifact. Maybe those are from the paper towel that I used, from the fibers coming off of them. That's my guess for that after I did the staining. So this particular one, not particularly good, but still, that's as good as we're going to get. And that's because these bacteria are not the textbook kind of shapes. And that's essentially not uncommon. So sometimes what we'll see is, for example, the slide that we just looked at, where there were a lot of rods, right, with some coccus looked like they were sticking out. Textbook, very clear, that's what a bacillus is. Some of these other ones, they just don't look like the textbook, and that's the way that works sometimes. So for the last slide that I want to show you, it's a slide that I put 2 types of bacteria in, 1 sample, and then I did a gram stain. Okay, so what we're looking at on the screen then is showing both different cell types. Okay, so what we see are the dark circles or spheres, and then we've got the red elongates. So my question to you is, how would you classify those? Just based on shape, what would you say we are looking at right now? Okay, so for example, what is this particular shape called? Right, so let's not call it a circle, and let's not call it a sphere. That is called that's right, coccus. So that's a coccus, that's a coccus, that's a coccus. Wouldn't you like to be a coccus too? Well, not if you're this bacterium. Right? This bacterium does not want to be a coccus. What type of bacterium is this? That is a bacillus, a very long, stringy bacillus. Okay, so that is what we're looking at there. Now on the screen, through the camera that's video recording us this right now, we can't actually capture this. However, when I see the colors on this screen, So I'm going to go ahead and open up the iris diaphragm to let in a lot more light. There we go. Some of the colors are popping now. And so what colors do we see here? Okay, so if we leave a little bit out of focus, I think that's the only way. So what color is this? That's right, that's pink. So is that gram positive or gram negative? That's right, gram negative. And these are purple or blue, so those are gram positive. And so this would be a gram negative bacillus. This would be a gram positive coccus. The fact that all these coccus are together, you might call that staphylococcus because that's what staphylococcus means. And so when I change the focus plane here, there we go, the camera that's video recording this ends up making that look purple. But Greg, would you agree that these look pink on the screen?
Yes.
Excellent. So that is showing you the gram classification of a mixture of bacteria where finally at the end here we see some textbook pictures of coccus and bacillus. And what we're looking at here, this is, for example, this is diplococcus because there's two. So this would be coccus, this would be diplococcus, this would be triplococcus. If you see a chain like this, that would be streptococcus. If you see a cluster, that would be staphylococcus. The fact that you've got a bacillus with a bacillus, that would be a diplobacillus. Since we've got bacillus after bacillus after bacillus after bacillus, that is a streptobacillus. And so what you would want to do is you would want to go ahead and snap that. And you would want to go ahead and save this and then put arrows and put diplococcus, coccus, diplobacillus, etc. And that's how you would go ahead and classify these. And so that will be, I think, the last video showing how to do the microscopy on the slides that you obtained from the company itself, as well as ones that you made for the end of the Ubiquities lab, as well as the aseptic techniques lab, and then you stained in the direct staining lab and the gram stain lab. And so it takes some time to do this. I hope you give yourself the right amount of time, and I hope you take the time to enjoy it.